Hi, this is the Year 7 cells topic. Now, to start off with, this is then the snaky diagram. This kind of shows the route through the unit. So it's the Year 7 cells. We've got microscope, animal cells, specialized cells, diffusion, respiration, unicellular cells, and then the end of the unit. Okay, it's important to know that route so you then know what's coming next. Right, and if you're smart, what you then do is you then have a look at what is potentially getting taught in the following lesson. Firstly, the microscope. Right, so what we've got, this is a very simple diagram of a microscope. We've got a number of different things that you kind of need to know. At the bottom here, you've got a light source. Right, usually what it is, it's like just a light that's got a bulb at the end of it, and what it does, it shines light directly through the stage. The stage is the part where what you do is you put on your microscope slide. The objective lenses, right, there's usually 1, 2, 3, right? What they are is they're then the lenses that you can kind of twist, right, to make the magnification higher. So you might have times 4, times 10, times 40. The focusing wheel, right? Quite often, again, you'll have two. You'll have like a fine focus and a coarse focus. The coarse focus is to get it into the right place approximately. The fine focus is then to get it as precisely focused as you possibly can. The last thing on this diagram here is then the eyepiece lens, right? That is usually times 10, right? What that does is then magnifies it 10 times, but it's the part that you put your eye to. Just for a bit of information, if you're using that lens there on the objective lens and you're using the eyepiece lens there, to work out the full magnification, you just times them together. So that would be times 40. And if it was that one, it'd be times 100. And if it was that one, it'd be times 400. Now there is a required practical, right? And the required practical comes in two forms. The first form is you've got a complicated version of a microscope. The ones on the previous stage are the main parts that you needed to know about. This goes through the method, right? 4 steps. Number 1, turn the nose piece to select the lowest power objective lens. So what you do is this part here, you turn it around until you got the smallest one pointing downwards. Turn the adjustment knob until the cells come into focus. Then what you're doing is you're doing this adjustment and you're doing this adjustment. I'll just change the colour. So this adjustment and this adjustment to focus it. Then the idea is then to make a clear labelled drawing of some of the cells. So on a piece of paper, you're then going to draw exactly what you're going to see. And often what it'll do is like a brick wall. On the diagram itself, write the magnification, right, and remember to multiply the objective magnification by the eyepiece. So if you're using an objective lens of times 100 and your eyepiece is times 10, remember you do 10 times 100 gives you 1000. Right, now the second part of the required practical is preparing an onion slide. Now you also are supposed to do a cheek cell, right, and it kind of works in exactly the same way. So what you do, you start off and you get an onion, right, and you kind of cut through the piece of onion, right, and then eventually what you'll do is you'll then be able to kind of get a pair of tweezers and peel off a really thin piece. Once you peel off that really thin piece, what you do is you get your microscope slide and you put a little drop of water on it. You then put on your piece of onion, right, which is 1 cell thick because you've peeled it off, all right. That then just goes onto the water, right, to then kind of hold it in place. Then what you do is you get some iodine, okay, so iodine, and you drop the iodine onto the onion. And what the iodine does, it stains the cell. Once you've then got your iodine on there to stain the cell, you get a cover slip, which is a really thin piece of glass. You put the cover slip over the top, press it very gently to get rid of the air bubbles. Sometimes it's better to actually leave these slides for like 24 hours because then the iodine then can stain for much longer. Now, microscopes themselves. Okay, so what I've got here is I've got light microscope and I've got an electron microscope. Right? Light microscopes are the ones that you get in the lab. Electron microscopes are very, very expensive and they'll often be at the universities. All right. Now, I'm just going to go through, right, some of the information here. So the radiation used, it uses light and that uses electrons. Okay, so a light microscope uses light, electron microscope uses electrons. Magnification, 1,500 times 2 million. Right, so the magnification on this is quite small. That is very big. The resolution. The resolution is how clear an object is. I'm going to give you a definition in a minute. So the resolution is we can see something 200 nanometers and 0.2 nanometers. So the resolution on this is very good, right? And on this one, it's actually quite poor, right, in comparison to others. Size of the microscope, they're small and portable, so that's definitely an advantage. And then a electron microscope is very large and not portable. A school microscope might cost £100. An electron microscope might cost £100,000 or even £1 million. Now, two key words that you need to know: resolution and magnification. So resolution is how clear the picture is. Think about your TV at home. You've probably got a 4K TV. Originally, there used to be 1080p, right? And then there used to be 720p, all right? And what the 4K means it's got much better resolution. There's more dots per centimetre squared. But resolution is the ability to distinguish two different points on a specimen, right? So the higher the resolution, the easier it is to distinguish between 2 points. Magnification is how much bigger an object appears compared to the original object. So if it's times 100, that means it's 100 times bigger. Now this is then the magnification equation, right, where total magnification is the eyepiece lens times the objective lens. So total magnification, if the eyepiece, the part where you put your eye to, is times 10, and the objective lens is times 35, what you do is you multiply them together. Eyepiece times objective, 10 times 35 is 350 times. Right, now onto cell labelling. Okay, now what I'm going to do is I'm going to go through the names of the different parts of the cells. Then what we're going to do is we're going to talk about the functions. Okay. So we'll start off with top corner here is the chloroplast. It is the green thing within there, right? It contains chlorophyll. Then what we've got is we've got this part here, which is the vacuole, right? The vacuole is again only in a plant cell, just like the chloroplast is, and it contains cell sap, but we'll do a little bit more in a minute. Around the outside, we've got the cell wall, right? That's the darker black layer around the outside. All three of them are only in a plant. These five, cell membrane, all the way around the outside, a bit like your skin. Nucleus is the big black dot. Cytoplasm is the white bit. Mitochondria is the blue thing, and ribosomes are the red dots. These five here are in an animal cell. All 8 are then in a plant cell. Now, functions. Okay, so what I'm just going to do is I'm just going to put a P there, I'm going to put a P there, and I'm going to put a P there, right? Because what they are is they are only in plants. Cell wall supports the cell and keeps its shape. It's strong, it's made of a substance called cellulose. Nucleus contains DNA and controls the activity of the cell, right? So what you've got is you've got your cell and there's a nucleus in it. That is then where the DNA is. Respiration occurs here. So in the mitochondria, that is then where respiration occurs. The vacuole is full of cell sap and it maintains its shape. So what it's got a lot of water in it. So if it loses water, what happens is the plant falls over. If it's got plenty of water in it, the plant is then upright. Okay, I'm going to circle that one green there as well. Then back to cell membrane. the cell membrane controls the movement of substances in and out. In a way, it's a bit like our skin, right? So our skin can control what goes in and out. Ribosomes, protein synthesis. I don't like proteins are made, right? It's protein synthesis. Cytoplasm is where the chemical, am I going to fit it in? Oops, chemical, there's a C there. Chemical reactions take place. And the final one, which again is a green one, is where light energy absorbed and exchanged or changed into food. And that process is called photosynthesis. Right, specialized cells. Got muscle, right? There's three types of muscles. There's skeletal muscle. There is cardiac, and there is smooth. Skeletal muscles are the ones that are kind of connected to your skeleton. Cardiac is your heart, and smooth is your intestine. However, the specialism for a muscle cell is the fact that it can contract. Okay, so the most important thing about any muscle cell is the fact that it can contract. It also has a lot of mitochondria, so that it can do respiration to provide energy. Then we've got a nerve, right? Now nerves, so these parts here, what colour should I do it? Oh, blue. These parts here are the nerve endings that could be in your skin. And what they do is they sense what is then going on. An impulse goes from these nerve endings down this orange part in the middle, which is called the axon, okay, and it gets to the end, which is then sort of the dendrite, towards the kind of the spinal cord or like a junction. Now, the way that a nerve is specialized, it is specialized by being long and the fact that it can conduct electricity. Okay, so this blue line that I've put on there is the movement of electricity. Sperm, okay, so I'll do sperm in red, right? Sperm has a number of specialisms, right? It has a tail for moving. Number two, it has a lot of mitochondria in the neck, right? That bit there. So what that can do, then it can produce loads of energy to make the tails move. Number three, cell is probably the smallest cell in the body. And number four, specialism, in the head, it has got half the number of chromosomes. Okay, and then the other half is in the egg. Right, and I'll just do a fifth one. It's got this stuff here, which is called an acrosome. And what they are is they are enzymes that help break into the egg. Root hair, right? So pretty much what you've got really is you've got your tree, right? It's obviously a tree, it's got branches coming off it, right? Looks more like an ant, right? And then what you've got across there, right? You've then got the ground and you've got roots coming off it. Now what you imagine would happen is you would imagine each one of the roots, right, is just where water goes in and minerals go in. But what a root hair does, okay, if I actually draw a root here, right, so that's a root and that's the ground, right, what a root does, it absorbs all the way around the outside of that root. That's where it can absorb the water. But what plants have done is plants have developed root hairs, right, which is this kind of big long nose bit here. These root hairs can also absorb water and minerals. So if you think now water can now go through there and it can go through there, what has happened is, and the main specialism is that the surface area has increased. So its main specialism is an increase in surface area. Oops, that's a very bad A. Right. Xylem and phloem, right? So what we've got is we've got root hair is one plant cell, then we've got xylem and phloem, right? So what I'm going to do is I'm going to go through xylem first, All right, and what I'll do is I'll draw my wonderful tree. There's a tree, there's the ground, there's the roots. Okay, it's obviously a tree. All right, xylem has a number of different jobs. It travels or transports water and minerals from the roots. It has no end walls between the cells. So this is what the cells look like, right? And what we've got now is there, the cells have actually broken down. So what it becomes, I'll write it over here, it becomes a bit like a straw, right? It's just like a hollow tube. It will only go in one way. It only takes stuff up a plant. And the final specialism is the cells are dead. So xylem, right? Xylem takes water and minerals from the roots up the plant. Okay? It's a bit like a straw because it has no end walls, and it only takes stuff up. Then the final thing is the fact that they are dead, right? So xylem cells are actually dead. Right, phloem. Phloem carries mostly, or the best example is glucose. So what happens is glucose is made in the leaf, then some part of a root, so glucose is made there in a leaf. That bit of the root needs some glucose, so glucose has got to go from there down to the root, right? And what they do is, to do this, is they use phloem. So it carries glucose. I'm just going to skip that next one first, right? What it's got two-way movement, which means it can take glucose and other substances up and down. The cells are living, but the most important specialism that any of them have is something called a sieve plate. So it's a sieve plate. And what they are is these things here. And what they are part of the process of deciding whether minerals and glucose goes up or down a plant. So in summary, phloem is specialised because it's got two-way movement, up and down. They are living, it transports glucose, and part of the decision-making process is by the sieve plates. Okay, now a definition, right? So what we've now got is we're now talking about diffusion. Right? Best example is somebody walks into the room with perfume on. The person closest to that person smells it first. The person furthest away smells it last. It can happen in a liquid. So if you look at that there, so what there is there's a line of purple particles at the bottom. Eventually what they'll do is they'll diffuse and fill the whole thing. This here is the perfume example, where you spray perfume here, and what happens is they all spread out by diffusion until the whole room is full. So the definition is the spreading out of particles of any substance in solution or in a gas, resulting in the net movement, right, from an area of higher concentration to an area of lower concentration without using energy. Now, that's quite a long definition, right? So what I would do is I'd kind of reduce it down a little bit, and I would then say diffusion is the movement of particles. Right, I'll just change my colour again. What colour shall I do? I will do pink. It's the movement of particles from an area of higher concentration to an area of lower concentration without using energy, which means it's passive. Aerobic respiration. Right, now this one's not completed, right, and this is then the word equation. So aerobic respiration is the process by which organisms release energy from food, right? So you take in your food, and what you do then is you do aerobic respiration to release the food. Okay, I'm going to do an arrow there, and this is glucose plus oxygen makes carbon dioxide and water. Glucose is your food, oxygen is what you're breathing in, carbon dioxide is what you're breathing out, and water is also what you're breathing out. Now that's the word equation. Now what I'm going to do now is I'm going to write the symbol equation. The symbol equation is C6H12O6, which is the chemical formula for glucose, plus oxygen, which goes to CO2 plus H2O. All right, so you're taking in food, which is glucose, you breathe in an oxygen, you breathe out carbon dioxide, you breathe out water. Now, sometimes what they might do is they might say a balanced equation. And to do a balanced equation, what you have to do is you have to put a 6 there, a 6 there, and a 6 there. Right. Now, in lessons, what you'll do is you get a bit more detail about these different things, right? But what we're talking about now is we're talking about unicellular organisms. And there are 4 main groups: amoeba, bacteria, euglena, and fungi.